Hi everyone. So in this week's lab, we are going to be focusing on carbohydrate catabolism assays. I gave you a lot of background information about each of those assays, and quite frankly, it takes a lot longer to explain all the science and biochemistry behind each of the medias that we'll be using, and it takes a lot less time to actually inoculate the tubes. So in this short video, I'm going to show you the different types of media that we'll be using so that you're prepared to inoculate all this media when you come into class on Tuesday. So we're going to start off with a liquid media that you know and love. You've seen this before. What you might notice is that there's a Durham tube in this particular case of liquid media. And that Durham tube is just a small inverted test tube that's placed inside of the media. This is new media. This is called semi-solid or semi-solid deep media. And so it appears to be solid media, but if you tip it, you can notice that it actually has a much softer consistency than the media that we use on the plate. So this has about half of the concentration of agar that our plates have. And lastly, this is called a slant. A slant has the same amount of agar as you will find in the plate. So if you touch it, it will have that bouncy resistance. But when the media is put into the tube, we let it cool at an angle. And that will create this broader surface that we can use to inoculate our bacterium. So this is a slant. And then you guys know and love the plate. So let's go through how we'll inoculate these different types of media that have various amounts of agar in them. So let's start off with the liquid culture. This will be a review for you, but I'm going to show you one kind of best practice when I'm inoculating this tube. So I have my nutrient agar plate that has a streak plate on it. I'm going to flame my loop to collect a colony. Remember that I'm going to want to cool the loop in an area where I see no bacterial growth, and I will collect one colony. I usually like to choose one that's kind of isolated away from other colonies. So this is the part that is new and sort of best practices. I'm going to use my pinky right here to collect the tube and take it off. I then turn it at an angle so no bacteria can kind of fall in there, and I do a swipe across the Bunsen burner to sterilize the top surface. I then am going to enter the tube with my loop and back and forth motion maybe 20 times. I can still see that the bacteria is on there. I'm not going to obsess with it because I don't need too much bacteria on there, but I went back in. Oh, one other thing, flame, and then the cap goes back on. And then before you put this back on the surface, you want to make sure to flame it to sterilize. Okay. Next up, we have the semi-solid media. So we're going to be using this for the OF glucose, and we're going to move away from using our loop, and instead we're going to use a needle. Not sure if you can see the needle, but it looks like the loop. It's identical to the loop, the handle, the metal that is used, but you'll notice at the very end there's no loop. It's just a straight needle. So to inoculate this media, we're going to, again, heat sterilize our needle, maybe about 3/4 of it. We're going to cool it, maybe the first ½ centimeter or so on the media in an area where there is not a colony. And you're going to do your best. It's harder than with a loop. You're going to do your best to kind of collect a little bit of a colony or all of a colony if you can get it of the bacterium on the tip of that needle. I find that I don't want to go straight perpendicular to the plate. Instead, I come to the side, and that allows me to get more of the colony on the broader surface of the needle. So now I'm going to use my pinky. I'm going to pull up the cap. I'm going to heat sterilize the lid. And now to inoculate a semi-solid, you want to go down into the media all the way to the bottom, and then you want to come up. Your goal is to have a single point of inoculation. You're not going to be able to make it perfect, but what you don't want to do is stir it around. You're not trying to mix up the media. Instead, you're just trying to stab it all the way down and all the way up. And again, my apologies, I forgot to flame the lid. And again, sterilize. So that is the semi-solid media. Remember, with the OF glucose, we're going to do this in duplicate, and then one of the tubes is going to get mineral oil on the top to create a less oxygenic environment, while the other one will have access to oxygen. So we can compare and contrast how the microorganism breaks down glucose in those two environments. Okay, next up we have our slant. Now I have to say this slant is very shallow. Sometimes you'll see the slant will take up maybe 40% of the tube. So they can look a little differently depending on the angle which you are cooling your media in. And so for this, we're going to want to make a zigzag pattern starting from the bottom and moving your way up. So after I label this, I am going to use the loop for this. Sterilize my loop. I'm going to cool my loop. I'm going to collect the colony. I'm going to use my pinky to take off the lid. I'm going to flame that, keep it at an angle. And then, this might be hard for you to see, but I'm going to start with a zigzag pattern, starting with the bottom and moving my way up. And maybe go back and forth, maybe 12 or 15 times. Flame. And now that media has been inoculated. Okay. Last, we are going to be using several types of media this week. That have different properties. So we'll have the starch media, we'll have the EMB media, which has this purple color, and we'll be using the mannitol salts and even the hemolysin. So you're going to be using all of these in addition to a nutrient agar plate. The goal of the nutrient agar plate will be to ensure that your bacteria is growing. And for all of these, you're going to be inoculating bacteria on these plates in the same way. So first, we're going to make a quadrant, labeled quadrant on the bottom, where you label each of the bacteria you plan to use. This is our mannitol salt plate. We are then going to use our loop, and we're not going to be conducting a streak plate technique. Instead, we want to have a small zigzag. I like to start from the outside and move my way in. And one thing that should be a focus is that you don't want the bacteria from one quadrant to move into and contaminate another quadrant. So I tend to do small zigzags so that I can ensure the bacteria grows separately. So here I can see my quadrant, I have my loop, and I'm just going to conduct a little zigzag on that surface. Of course, you're going to want to heat sterilize your loop between all the bacteria. You might even choose to label all of your plates, for example, down here, and then inoculate E. coli in each of the different types of media to be more efficient. And so we'll be looking at, as you know with these plates, we'll be looking at growth of the bacteria, whether the bacteria grew, for example, with this mannitol salt plate, or whether it didn't grow. And then what you'll also notice is that the appearance of the bacteria will sometimes differ. So we can see in this case, the bacteria is creating a yellow color within the media, which is meaningful. Over here, we can see that in the hemolysin plate, the red blood cell plate, we can see that sometimes the background is gray. And if we put this under a light box, you would see sometimes we can see clearing, which is going to be important in determining what the role of the bacteria is. In our EMB, we see the lack of bacterial growth up here and here. And then we see bacterial growth that almost has a metallic-like quality, where over here the colony still appears sort of a off-white color. So we'll have a lot of fun this coming week, and lots of color changes, lots of inoculations, and I look forward to seeing you all.